Next along that pathway of shipping things outside the cell is the Golgi apparatus. And I really think about the Golgi apparatus as the post office, or the mail system, because basically what it's going to do is going to take proteins that were made over here in the endoplasmic reticulum, then put in a vesicle and shipped to the Golgi. It's going to take them, modify them, and it's going to bundle them in these vesicles that are really destined to go to that cell membrane. They're either going to fuse with that cell membrane, or like you see here, they're going to dump something outside the cell. This is pretty important because without the Golgi apparatus, we wouldn't have any way to load our UPS trucks or our vesicles. And that would be a problem, because then we wouldn't be able to rebuild cell membranes, we wouldn't be able to ship proteins to cell membrane, and the functionality of the cell would be really, really hampered. So the Golgi apparatus, I think of it like a shipping center, and it's pretty important. If we look at the apparatus itself, it really, like I said, acts like a shipping center, and it has two sides to it. This side right here is the cis side, and this side is the trans side. The cis side is your receiving side, and the trans side is your shipping side. And so typically vesicles will move right through in that direction, and while they're in this spot right here, that's the location where they're going to get modified. And again, it's going to modify, store, and package proteins and other materials from the ER for either storage in the cell or release outside the cell. That's the main function, and because of that, we draw the parallel to the post office. Some organelles that capture or release energy, at least there's two main types that are important, and you'll notice that in animal cells we have these little guys right here called mitochondria. They're the powerhouse of the cell. We'll talk a little bit more in a bit. But in plant cells we also have mitochondria. Plant cells have those, but plant cells have in addition these little green structures called chloroplasts, and those are absolutely important because they're going to be the site for photosynthesis. So let's talk about chloroplasts first. They're biological equivalents to solar panels, and that they've got these pigments on them that collect sunlight and actually generate an organic carbon. And this is pretty awesome because they're going to take carbon dioxide, which is non-living, and they're going to use sunlight, which is the solar panel analogy. They're going to use sunlight to take that carbon dioxide and turn it into C6H12O6, more commonly known as glucose. If you take two of those glucoses and put them together, you get sucrose, and that's your table sugar that you would put on your Frosted Flakes. This is pretty important because they end up being pretty much the basis for most energy in our ecosystem, all coming from these chloroplast cells. And these chloroplasts are part of what we encompass into our endosymbiont theory, and that these chloroplasts have their own DNA, which is kind of strange because in eukaryotic cells we keep all of our DNA in the nucleus. But because these guys have their own DNA, we kind of theorize that they were once prokaryotic organisms that kind of got engulfed and have a symbiotic relationship with eukaryotes. Again, there are two membranes, and if we look at this particular graphic, there's a couple key things to note here. This little open space is what we call the stroma. And so the stroma is it's where the actual Calvin cycle takes place and the glucose is made. Other structures that are notable is this little guy right here is thylakoid. We stack a thylakoid in granum, so that right there would be the granum. And inside each one of these is actually where you find your chlorophyll. And so chlorophyll is the main pigment that's going to absorb sunlight. So that's the basic structure. This is a transmission electron microscope image of what a chloroplast looks like. Mitochondria are the powerhouse of the cell. Again, we're using the same image of a transmission electron microscope of a plant to show you where the mitochondria is at. They're the powerhouse of the cell because what they're going to do is they're going to take carbohydrates, they're going to take sugars, and they're going to completely break them down. They've got two membranes. They've got an inner membrane, as you can see right here. It's kind of folded on itself to help increase surface area. And then they've got an outer membrane. Again, just like a chloroplast, these guys have their own DNA. And that makes us think that they were probably their own organism, and a prokaryotic organism at that. But they've been brought in. There's some kind of symbiotic relationship between them and us, and we actually work together. It's theorized that mitochondria have been with eukaryotes longer because they have less DNA that's left to them than chloroplasts. They rely on us a little bit more. But these two membranes actually will build up this gradient of hydrogen ions, and as they move across, it's going to generate ATP, which, if you remember, we're going to talk about a little bit later. But this ATP is pretty much like what I call the $20 bill of the cell. What it's able to do is just buy stuff. So the more ATP you have, the more stuff you can get done. And ATP is primarily made in the mitochondria. If we talk about cell boundary, there's two main types. Here I've included a prokaryotic cell because, like I said, prokaryotic cells do have some of this stuff, and they do have cell walls. And I've also included a plant cell. You notice that there's no animal cell. And that is because animals don't have cell walls. There's a major difference between the cell wall of a plant and the cell wall of a prokaryote. For instance, prokaryotes, their cell wall is primarily made up of peptidoglycan, and in plant cells, it's usually made up of cellulose. But it produces the same result, and the cell wall is an actual strong supporting layer that will give the cell a rigid structure. And so it typically means that these organisms don't move very well, especially in multicellular organisms like plants. They don't have any motility at all because of that cell wall. In some prokaryotes, you'll notice that this flagellum is hanging off here. That flagellum can help propel the prokaryote around. It kind of acts like this thing's a little submarine, and that's its little propeller. You'll also notice that there are some small cilia here. Both of these structures, the cilia that I highlighted there and this long flagellum, are used in motion of a cell. So they'll whip back and forth. They're connected to the cell membrane and the cell wall. And that cell wall, again, is a supporting structure that kind of gives the shape of it. Cell membranes are universal to prokaryotes and eukaryotes. We find them in all living organisms. And basically they contain a way that regulates what enters and leaves the cell. That's the key role of a cell membrane. It's like the bouncer of the cell. It decides what's allowed in and it decides what's allowed out. If we talk about the basics of it, though, it's really made up of these lipids. And this shape is actually very representative of what they look like. We have a hydrophilic head, which is a water-loving head. And so that hydrophilic head actually likes water. And then we have hydrophobic tails. And these hydrophobic tails, they don't like water at all. And so what ends up happening is we'll form this phospholipid bilayer. So if you look, we have one layer of lipids here, and we have a corresponding layer of lipids there. And so what we have is any H2O, or water, that's out here is perfectly happy. No problems with water out here. But as we get towards water trying to be in this area, that's a major no-no. This area that's right here is our hydrophobic zone. And so that's really how a cell membrane keeps things separate, is that water doesn't want to be in here. And so anything that wants to mix with water, which is quite a lot of stuff, can't go through there. If you watch the video on cell transport, we'll talk a little bit about these membrane proteins. And they're pretty important because a membrane is what we call a fluid mosaic. And so if you look at it, it's made up of all these different lipids, and they're in a phospholipid bilayer, so it's, the phospholipids in two layers. And then they've got these proteins put in here. For instance, this protein right here is what we call a glycoprotein, and they're used in cell-to-cell recognition. So when an individual needs a blood transfusion, we're going to pay particular attention to what their blood type is. Because if you give somebody who has blood type B blood type A blood, their cells are going to recognize this glycoprotein that's for cell recognition, and they're going to kill that cell. It's not them. They know it's not. And so this carbohydrate chain attached to your protein can have some pretty important stuff. And there's some other things that are happening here. We have some carrier proteins, and this particular one right here is important because it's an aquaporin. And so I hope you can see that this plasma membrane is really just kind of made up of all kinds of good stuff. And that's why we call it a fluid mosaic model. Fluid meaning they can move, and that's why animal cells really don't have cell walls. They just have a cell membrane because, well, they need to move. And so all of your cell membranes are critical for keeping things in your cell that should be in it and out of the cell that should not be in there. And they're able to move. And so what we actually see is that these little phospholipids, they can switch places left and right. They do that pretty often, but they very, very, very rarely flip-flop. So if you're talking about flipping from one layer to the other, that does not happen very often. It's very, very rare for that to occur. But they do move side to side. So if we were able to come in here and, let's say, tag one of these and then came back and looked at it again, it might be way over here. That's not uncommon. And it'll move around really well. There's a couple different things that can happen with these lipids. For instance, these tails could be saturated or unsaturated. Saturated means you can pack more of them together, and unsaturated means you got this little kink in this one, which is actually a double bond, and then they don't pack so well together. Certain cells in specific environments will actually want saturated. Let's say, for instance, it's a very, very warm environment. Having a lot of saturated phospholipids will help you stay in one piece and not melt. But if you're in a very, very cold environment, having a whole lot of saturated phospholipids will make you solid, freeze solid. So most of those cells will have a ton of these unsaturated phospholipids, which will have a double bond in one of their lipids that kind of cause it to stand out like that. So this cell membrane is absolutely important. What it really does is regulate what moves in and out of the cell, and it's what we call a phospholipid bilayer. Two layers of these phospholipids that is in a fluid mosaic model. We have multiple different things in there. It can move around. It's absolutely important to cell function.